Started. Hello, everyone. Welcome to our June session of the Center for Reproducible Biomedical Modeling seminar series on tools and techniques for modeling biological systems. Today, we'll hear from Dr. David Nickerson on an annotation library for biosimulation models, but we hope you'll also join us next month to hear from Jin Xue on an SBML network viewer, and also from Matthias Kunig on his work developing SBML utils. For our seminar guidelines, we'll have about a 40-minute talk by Dr. Nickerson today, and then we'll have a group discussion following that of about 20 minutes. Our goals are to identify common challenges we all share in our modeling projects, share our personal experiences and solutions with these sorts of challenges, brainstorm new solutions together, and ultimately create a collaborative community of modelers. Our guidelines today would be to err on the side of commenting during the discussion section. If you have a question, it's likely somebody else has the same question as well. Honest debate is encouraged if you might have dissenting opinions from those suggestions that are offered. And just mute your microphone when you're not talking during the session so that we can hear those who are engaging in the discussion or giving the talk. So today we welcome Dr. David Nickerson. He leads the Auckland Renal Physiome Project, as well as the Physiome Model Repository, and helps lead the Physiome Journal as a senior research fellow at the Auckland Bioengineering Institute in New Zealand. In addition to his group's research work, he is committed to the work being done in the Combine community. He serves as a member of the SedML editorial boards, and is also a coordinator for the Combine conference, which ultimately helps the modeling community develop shared tools and standards. Dr. Nickerson also serves as the co-director for the model simulation and annotation team at the Center for Reproducible Biomedical Modeling. And in this role, he heads a model curation service, which enables reproducible model dissemination. And he has also helped lead the production of a library to annotate biosimulation models with distinct biological meaning. And today he's going to share more about this annotation project. So welcome, David Nickerson. Dr. Nickerson, you have the floor.
Cool. Thanks, Veronica. Thanks for the introduction, and thanks for rescheduling the seminar at a slightly friendlier New Zealand time, which is good. And just, why didn't that work? Share. Okay. So hopefully you can see the slides. So today I just wanted to briefly sort of run through this work we've been doing with what we call libOmexMeta. And it's really about how we can help harmonize model semantics. And for this talk, when we talk about model semantics, it's really about the biological meaning of a computational model. There's obviously a lot more semantics around, but for today we'll just focus on the biology. And so, this work is obviously a group effort with these people, and especially Karen, who is the main developer, who's implemented all this lovely code that we're going to play with today, and working very closely with Anand, John, Herbert, and Max, both in Auckland and at UW. And it's a couple of links, and we'll pop them up again as we go through. And if I can click in the right place, there we go. So the goal, as I said, is really, how do we harmonize the way we describe our models? So a few years ago, a group of us came up with this paper, which sort of lays out how we should do that. And so the library I want to talk about today is really, getting to the technical details of how we can do that in real life. And essentially, what we want to do is make our models more fair. So if we can all annotate and describe our models in the same way, it makes things more findable. If we can search over multiple different sources of models in the same way and find useful information. I think that makes models more accessible if we can all understand each other's work, which we can do if we're all sort of using the same way to describe our models. And then obviously more interoperable when software tools can also understand model semantics as well as the sort of model itself. And then I think all of that together helps improve reusability and obviously reproducibility. And then, you know, this is all good, but then it's not easy. This is not an easy thing to do, right? So if we have a nice implementation that everyone can use, that everyone can integrate into their tools to provide this sort of core technical implementation, then that helps the different tools that people actually use in their day-to-day research to adopt these sort of more consistent annotation practices. And that's really where libOmexMeta comes in. And as I said, you know, we're focused here on models and more specifically describing the biological semantics of models. But in general, this sort of approaches that we'll show today can be applied to many different things with various sort of extensions or adaptations of the existing code. And perhaps in the discussion, we can get into some of those directions. So just a bit of background. I'm not sure if everyone's familiar with Combine. There's the logo and there's the website with lots of information in there. But really, you know, the core of Combine is a set of standards that we adopt to try and represent different types of information, different types of knowledge. And for what we're talking about today, we're really looking at this sort of modeling level, and in particular, the SLML and SPML standards for encoding models, sharing models. But the other one that's quite important is this little one here that doesn't really show anything, but essentially this idea of a Combine archive. So some way where you can package up all the different resources to do with a particular study and make it available for sharing and reuse. So the Combine archive is a sort of standard way to do that. Essentially a zip file that has all this information, all these files, and what we're doing with libOmexMeta is you can then add these sort of semantic details through the annotations. And essentially this idea of having a sort of package or an object of some kind that you can share, the Combine archive is, you know, a way you can do that in a file, and different tools and different repositories have their own sort of representation where you can do this. It typically is interoperable with this idea of a Combine archive. But essentially the main idea of having a way to package up everything, keep it together, and to try and help synchronize the different resources in a particular study. And then, you know, this idea that's mainly led with Henning at the EBI and the biomodels team of collecting, you know, all the semantics and the sort of meta repository called Model Exchange, so that you then have this place where you can search. And this is based on this idea of proteome exchange, which already exists, where they found it was very successful to try and link all these repositories of proteome knowledge together in a way that lets you search and discover. And so what we're trying to work towards is how we can do this for models, and then give this sort of one place where you can go and find useful models. I can go much quicker than I thought, so I'll try and slow down. But just to give an example of how this might work in practice, a student of mine here at the ABI, Yuda, has been working on a project looking at NLP and how we can use that to search for models. So here's a nice little demo, and you can go there and you can enter sort of natural language queries. And then what you can do is in this demonstration, we've extracted the semantics from two repositories, biomodels, which is mostly SPML models, and our physio model repository, which is mostly SLML models, by trying to use these sort of common semantics to find things. And so what you can do in this tool is if you search for something like hexokinase activity, underneath it's turning it into a bunch of sort of database queries to try and find things. And for a fairly generic term like that, you get lots of results, both for biomodels and for PMR. Right, so you can see lots of different things. You're able to start linking to the specific things within models and seeing what they are. There's just a benefit of annotation in general. But then what you can find now is if you search for something much more specific, such as hexokinase activity with ATP as a reactant, you generate different set of queries that are getting executed. And now you find a sort of much smaller subset of results that are much more specific to the query. Searching across, in this case, there's nothing in PMR that has this information, whereas there are a few results from what we extracted from biomodels. So I think that sort of starts to show the sort of power of what we can do once we do start annotating our models in the same way, makes these sorts of queries that can then work across different repositories possible. And then just to show a very different type of example in a different project I work on, in the Spark project, where we're looking at how to map between experimental data and models, essentially. So you can see in this example shown here, we're taking, imaging data where people have segmented out. In this case, they're looking at the neurons in the heart. We have a model of the heart and how we can, essentially fit this model to the specific data, registering this data then to the model to bring this into a common framework to then integrate with other types of data or other different sources of data. And essentially, apart from perhaps having some cool pictures, this is showing how this type of annotation, in this case, it's a different annotation. We're looking at how we annotate anatomy, but essentially to do this process in a largely, semi-automated manner, what the investigators are doing is they're annotating their segmented data with anatomical terms. We're annotating a sort of organ model of the heart with the same terminology, using the same terms, so that then the software is able to match, between the data and the model to do this type of thing. And just to see if I can break things. And of course, it opens in a different window. If you did want to have a play, essentially this website that we're putting together under Spark is trying to bring all this together, driving a lot of these things through this type of annotation, where it's using these sort of common semantics to link all these things together. So if you want, you can go to the spark.science website and have a play. And it takes a bit to load, but essentially you can see the result of that mapping any second now. There we go. So you can see, and this is all driven by the semantics, and through this portal, you're able to essentially dive down and get the data and see and dive into those annotations if you're really keen. But that just, showed you it's a slightly different level. It's a different type of thing where we're looking focused, at anatomy, but how we can link all these things between, data and models by using common semantics, which I think, if I can get my slides back, I think really starts to show the power of how, when you have these common semantics, things will work. I've lost everything on my machine. Okay, so so this is great, and this is really useful things, but how do we make this happen, right? And this is really where libOmexMeta comes in. So again, there's the GitHub site for those that are interested. And essentially, this is a C++ library, and it's aimed not primarily at the researchers or at the scientists doing the modeling directly, but more at the people who are creating tools that the modelers use, or anyone uses when they want to access models. So it's really providing the sort of developer level support for reading, writing, editing, and managing these semantic annotations. And then within Combine, we have what's called the Omics metadata specification, which sort of defines the way we should do these harmonized annotations. And so libOmexMeta is really aimed at supporting that specification. And then because a lot of people like Python, there's a Python front end to the library, which thanks to Karen's hard work is really as simple as pip install pyomexmeta. And it generally works on Windows and Mac and some Linuxes. So under the hood, essentially the library, sorry. No, I don't know what's happening. Keep away from my mouse. So essentially under the hood, this is all about reading and writing RDF. Typically in Combine and Combine modeling standards, we use RDF XML, either directly or embedding it in other XML documents such as CellML or SBML. But the library itself, thanks to using this Redlin suite of tools underneath, supports various formats. And we typically now would use something more like Turtle, especially in documentation and demonstrations. But all these things are available because we're using these sort of standard W3C based semantic web tools. And then, within the library, we can expose things so that you can query the annotations using the Sparkle query language, which again is a standard sort of query language for these types of annotations, or looking at how we can visualize the graphs using various tools. Currently it uses GraphViz at the moment, but I think Karen's been looking at other options that are a bit more portable. So as I said, the real capabilities of libOmexMeta are this ability to read, write, create and edit annotations, and also looking at how we can extract annotations from existing models. So we have huge repositories full of models. A lot of those models are annotated, and in standard like SBML, where some of the semantics are encoded in SBML itself, we want to be able to extract all that information, all that knowledge, and make it available in this sort of common way, standard way, to make it more reusable, more findable, and more accessible. We can also extract annotations from URIs, and then working with another library under Combine, this libcombine, that really works directly with these archive files. You can see how we can then link these together to work directly with the Omics archives. So again, there's a GitHub page. From there, there's also a link to this sort of live demonstration example that we have that'll fire up a Jupyter notebook on Binder. And I'll get to that a bit later. Okay, so now I think that's enough background. So just to dive into the tutorial, as we go through this, I don't know if people follow along or anything, but feel free, I think, to ask questions as we go. If anything comes up, happy to interrupt as we go. So essentially, this part I'm going to try, hopefully, to demonstrate how you can use the Python front end to libOmexMeta, but you can imagine very similar sort of interface for the C++ or C interfaces. I'm doing this on a Windows machine with Python 3.9. I like to use virtual environments for everything. And if you do want to follow along, you'll hopefully be able to. But if you don't want to run it locally, you can go to the live demonstration site, and it's pretty similar to what we're going to go through in this tutorial. And if, like me, you want to copy and paste code rather than type it all in, there's a link here with this QR code that will show you the actual script that we're going to go through for anyone who wants it. So I might just leave that there. and try and find all the windows that have disappeared. Where's it gone? Okay. So essentially just to run through the whole process, so if we want to start, we can make a folder that we're going to work in. Right? And then because I like to keep everything in virtual environments, just to make sure I don't destroy my system, because I'm not the best programmer. And on my system, this is how I make a virtual environment. If I could type. And so it's just going to make me a nice, fresh, clean virtual environment. I'm using my system Python 3.9. Get that going. Oh, thanks, Veronica. And then, as I said, if I just pip install pyomexmeta, right, then hopefully that's going to go away and find things. There is a fair bit to install, so while that runs in the background, I can just pop over here. And of course, it's another different one. Bring that up. And so here you can see this demonstration one. If you just click the launch binder option, it will then take you to this binder service that we use and fire up an environment that has this notebook in it. Depending on how people have accessed it and things and where the virtual machine spun up and things like that, it takes a bit just to get going. But if a few people load it, then it hopefully gets quicker for everyone else. But essentially, once that comes up, you'll see a notebook, a Jupyter notebook here, just called Notebook. And if you open that, you should get a pretty similar environment, or the same environment as what I'm getting locally, with this notebook populated with largely the same material that we're going to go through in this tutorial. And if we look back here, we can see it's just finishing up. So while it's going, if I now fire up that notebook, you can see, here it is, and there's a bit of documentation to help guide you through. And so feel free to have a play with that. Okay, so now we have that. I was just going to use a Jupyter notebook like that demonstration to show. So I just want to install, if I could, just install Jupyter notebooks, just to make the demonstration a bit easier. Again, it just takes a little bit of time to fire up. It always goes much quicker when I test it. But essentially, this is now going to give me, in my sort of environment, I'll have pyomexmeta and able to fire up a Jupyter notebook. While that's finishing, just to answer your question, Jim, in terms of using the natural language processing to Sparkle query to also look at using that to actually annotate models. I agree that the searching and annotating should use the same core tools. We haven't actually done that, but we do use existing annotations in the repositories to guide the NLP. And I think in the center, we are looking at how we can use that to also infer annotations and help define annotations. Okay. So now I can fire up a Jupyter notebook locally, and it never opens where it's supposed to. Bring that up here. Okay, so now I have a Jupyter notebook running. Just create a new one. And if everything's worked, I should be able to simply, I'm hoping that's big enough if I just maybe make it a little bit bigger to make it clear. And that didn't fail. So that successfully installed pyomexmeta. And this is the sort of typical import that we would use where we want to import this RDF for the RDF graph and these URI types that we'll get into a bit later. And so essentially, yeah, this should work in Colab as well, and various other things. And we're using Binder for our demonstration just because that was easiest, free, and doesn't need anything to sign up. But there's other various ways to host Jupyter notebooks online that should work. Okay, so just to create an empty IDF graph to show where this is the sort of thing we do, where we're making our IDF graph, because we're aiming to work with these omics archives. So typically, what we want to do is set the base file name for our archive, so that when we create annotations, we can always do absolute URLs within the archive, which again makes interoperability much better by having absolute things. So no matter what system you load the archive on, you always get the same RDF graph. And typically also set the model that you might want to work with. You can do things we normally do just to print, and then you can see the existing current state. And so here you can see these sort of namespaces that we're defining, where we want to set sort of a root one, which is a sort of dummy URL or domain that we use just for this purpose. It's defined in the spec to give us a sort of common base, and then this local one, which is essentially an RDF file within the archive, which we're going to use to define all our locally defined annotations. And then, if you want to, so by default, it does everything in Turtle. And if you wanted to, you could easily use other formats, just like in triples. In this case, there's no data, so your graph is empty. Yes, and to generate OWL or oboe, that's one I can't answer, I'm afraid. Maybe. But I think, yeah, sorry, I'd have to check exactly on those syntaxes. And then again, this is Python, and things are kind of accessible, so you can sort of explore the objects and their API. I'm just using standard Python methods. The documentation in the Python is now all in place, I believe, so various IDEs and things should help with the prompting and auto completion and things like that. Scene running out of time, so I'll start speeding up again. But essentially, just to go through the different types of annotations we can do with this library, before we start anything, we just need to define a couple of models that we want to work with. So in this case, I'm just defining a sort of simple dummy SPML model and an even simpler SalamL model for us to play with. And then what you want to do is essentially create an editor. So here we're going to, in this RDF graph, we're going to make an editor based in this case on this SPML model. And there's a few things like this semantic extraction where we can get the library to extract the semantics of any sort of existing annotations and structure in the model, which we won't get into today. And again, you could explore that with the Python tools and things like that to see what the API is if you wanted to learn. But in this case, jump in. And so what we want to do is add an annotation of the model to say who created it, and add this taxon to identify the taxonomy or the species for this model. And so when we do that, we can then print the RDF graph, and we now see we have an annotation. In this case, oops, I didn't set things up very well. And so it still thinks that this is my model file, even though that's clearly wrong. But so this is saying something about the model file, which we recently learned is wrong, but will be fixed shortly. But we're saying, it has this taxon. It's used its internal knowledge of what a taxon is to generate the full URI to use, so it's nice and unambiguous. So now if I was to explore and see what that is, and something broke overnight, there we go. You can see, it's essentially a cow, I think is what that is in English. And again, it's taken the ORCID ID and uses the full URL. So it's nice and resolvable. And if we go there, we hopefully see it works. It should be myself, which it is. Okay, so that's there, and then maybe you want to add more, so you can add another creator, and so on. And then so for some of these there are restrictions. So for example, the library expects ORCID IDs to be used to identify people at this point. So whenever you specify, you can either specify the full one or just the ORCID ID directly, and it correctly works out what it should be. If you try giving it something else, like a different directory entry or something, it tends to not be happy. And then another thing that's quite common that you want to do is to perhaps identify a curator to say who curated this model, add a link to a PubMed for a publication, and some information about when it was created. So now if I do that, you can see it's adding in this link to the publication. And again, it's expanded the PubMed ID to the full URL, adding in the state. And now when how we interpret a curator is essentially the person who's annotating the model. And so now, rather than being an annotation on the model, that is saying that person we just identified is the creator of this annotation file that we're working with, which again, we defined that file name earlier. Okay, and then as I mentioned, you could draw the RDF graph if you like a more visual way to explore it. Unfortunately, I don't have Graphviz installed on this computer, so I can't demonstrate that. But if you did, essentially it's as simple as that, and it would just work. And here it's just going to say, hey, we can't find Graphviz. But that is a nice simple way to then visually see your RDF graph. So that's when you want to annotate things about the model as a whole. The other thing that might be quite powerful is you just want to say something. So you want to make a singular annotation. So again, here we can just see, yeah, GraphViz is cool. What we want to do is just essentially specify the full triple. So in our editor, we're making a singular annotation, and for a full triple, we need to say what it's about, what the predicate is, and what the object is that we essentially want to annotate. And so now if we do that, you can see that it will generate this expected thing. It's about this entity. In this case, it's in the model with this predicate linked to that identifier. These hopefully all resolvable URLs. And then there's other methods if you want to do some other sort of more RDF specific type annotations. Okay, so but then the real power, now I'll really speed through this with the library is we want to make more detailed statements about things in our model. So we want to use what we call composite annotations, where we want to make an annotation that is essentially stating many things about a particular model. So now, and so the basic one of those, if we just make a new RDF graph to clear out everything we've done. And so the sort of simplest composite is what we call a physical entity, right? And so that's, you know, saying something about the physical entity itself and the property that's being simulated in the model. And so in this case, what we want to say is this thing, and here we use this URI type to explicitly say this is something in the model. And you know, so this is an identifier in my model. We use this model to say it's in the model, and I want to say it is this bqBio term. In this case, that's glucose, and I want to say that's part of this Go term. In this case, the Go term is cytoplasm, and then I'm using the OPB to say it's a chemical concentration. So essentially what I want to say is this thing in my model represents the concentration of glucose in the cytoplasm. So then if I do that, I get an error. I go, why do I get an error? And essentially what this then tells me is that, hey, the model that you've defined for this editor where you're doing this doesn't have something with that ID. Which is quite good. It's checking my model to tell me I'm actually annotating something exists. So that's good. It helps me make sure that I'm kind of making sense. So now if we go back up here and actually paste in a model that does have glucose, and then rerun this, then it worked. Sweet. Okay, and then maybe what we also want to say is something about glucose, but now in the nucleus. So it's a similar sort of annotation. And again, if I print out my graph, you can see now I have this. And so what you can see is these are the, you know, here we're saying these are the things in my model. So those are annotations against the identifier from the model, saying it is this bqBio term and is part of this Go term. But what we're missing is what's the relationship between these things. And so that's where the library now has defined these, what we call local annotations, which now are in this local namespace, where we're saying, you know, that this thing in the model has a property, which is this OPB term. So that's where the relationship between these things now comes in. Okay, and so that's an example in SBML. And now if we look at a similar example in SalamL, right? But now in SalamL, instead of SPML, we have variables which represent the physical properties, but the biology, the biological physical entity is more implicit. So here in this case, what you can see is when we make our physical entity, we're defining a new local thing with this identifier. And what we want to say is this thing is this, in this case, it's a piece of anatomy. I'm using FMA. So we're saying here it's a portion of blood, and it's part of this FMA, which is the lumen of the left coronary artery, and it has property, this thing. And now what we're doing is we're saying that this is the thing in my model. So here I have a model URI, the identifier in the model, and now I put the OPB term, right? So which is fluid volume. And so now I get slightly different thing where now you can see that the sort of entity part is in this locally defined identifier, whereas the property is what's in the SalamL model. Okay, and then if we jump in, and really running out of time, sorry. So if I just quickly throw up this example. So now I'm defining a new SPML model, has this reaction. You can see here's the sort of reactants and products. And what I want to do is define that reaction in the semantics, right? So it's a physical process. Again, here I have a reaction in my model, so I can talk about that in the model. It's a version of this go term, which is this thing. And now I can define, it has these sources, which you can see here are the reactants, has these sinks, which are the products. Those are all things in the model, so they all have model identifiers. In this case, it's stoichiometry, nice and simple. And there's a mediator, again, in the model. And has this property with this OPB term for chemical flow rate. So now, if I look at the generated RDF, you can see here's a listing about that reaction. It has this mediator, these sinks, these sources, and is this version of this thing. So again, now you can see it's defined internally, these sort of local things that we're referring to, where we can then expand on the definition and the semantics to show, additional information that it has multiplier and has this entity reference to this thing in the model. And so that gives you a nice sort of clear description of that. And then just to contrast that to an example in SalamL. Right, so if you're not aware of what's with the difference in SalamL, so SalamL, we don't have the reaction, that sort of biology defined in the language. It's just the variables and the math. So now when we want to describe that type of physical process, as we saw earlier for SalamL, we need to define these physical entities that we want to talk about locally. All right, so here we want to essentially talk about a different model, and now we're going to define those physical entities. In this case, we're talking about the cytosol and a hepatocyte with this one, and again, we're defining this locally. And then we're talking about other things to essentially build up the same type of reaction, but now we're talking about it and defining it all locally. And then, here we want to define that reaction rate. And as we saw earlier, now it's where, down in this has property where we can actually now link to something in our model, right? So this is where we have the model URL. And so now if we look at that, essentially, depending on how familiar you are with RDF, you can look at this, and it's a very similar graph in RDF sense to what we have here for the SBML model. But the only difference really is, which bits of it link to things in the model that we have, and which bits are in these sort of locally defined entities. So here, all our sources and sinks are locally defined, and it's this reaction rate. That's the actual thing in the model that we can talk about. But in terms of RDF, and if you were to look for this, the same information is all there. And so that means when I search, it'll find, using the semantics, I'll find this and I'll find the other one. Whereas if I was just looking at the CellML or the SBML directly, I'm going to see very different things represented. And then I think just because we're out of time, basically, the third one is, for the third composite, is it's looking at energy differentials. And that's particularly useful when you're looking at things like membrane potentials. But that's a very sort of similar type of composite. And so perhaps I'll stop there and leave a few minutes for discussion. But thanks for everyone who's followed along.
I think the tutorial helped make it a lot more accessible. So thank you for presenting it in that format. I think Dr. Sluka had a few questions. I don't know if you answered them, just about where the list of predicates came from, whether they're hard-coded or if you used a relationship ontology for them.
So I'm pretty sure we did not use the relationship ontology to get them. There are some hard-coded, especially these ones that will have a bit of smarts in terms of what they expect to be given, and so they are relatively hard-coded. I think.
Well, just to chime in, David.
Yep.
They come from bqBio, really, right? So part of and as you can see below in the code, is bqBio is part of, which is the combined subset of the relations ontology that they use. It's a very small subset, like 3 red predicates or something, but those are pretty much all that we use. I think the relations ontology in general has a lot more detail than we need. So I think I'm happy with the bqBio. Now, there are some limitations to the bqBio set. And if, for example, one of the relations that's important for reactions is the has reactant and has product relations, which we call has source and has sink. And so those we created our own namespace for. Yeah, there they are. So that's where those relations come from. And we're working with Combine to sort of expand the bqBio set to include those.
If I can butt in, so it sounds like it would really be nice if there was an interface, like a web-based interface to this, because actually going through and issuing one of commands in Python is, 1, painful, and 2, it's going to be incredibly error prone, and 3, takes a huge amount of expertise. I had put the comment in there, is it contained in or part of? Well, actually blood is contained in, it's not part of. Right. And those are subtleties that once somebody figures out what the correct one is, no one else should ever think about it again, right? Well, it should, there should be a question, is this intrinsically part or not intrinsically part? And even little things like you're annotating a species in SBML, well, it's probably in bqBio or in a protein ontology. So is there some way to provide hints that maybe you should look here? 1, because that way somebody doesn't wade through 100 different ontologies. And 2, it kind of enforces some regularity so that a person doesn't build a model and annotate one species with bqBio and then another chemical species with a mesh term. Right. Estradiols in mesh. But boy, you lose the context of what that thing is if it's annotated as from mesh instead of from
Yeah, absolutely.
bqBio. And those are the kind of, Kopasi has a kind of an interface for doing annotations, but it is really dumb. It's a species, and okay, what ontology you want to use? And there's a drop down with 150 entries in it. And maybe you should know that, well, this is a species, so at the top of the list we'll put bqBio and gene ontology or something.
Yeah.
It's probably one of these, but if you really are not sure, here's go find it.
Yeah. So just to jump in, sorry, David, but this speaks to sort of the work that Max Neal and I have done with an annotation tool. Now, the software that David's presenting today is not an annotation tool. The software is a library for annotation tool builders.
Right.
So in SemGen, our annotation tool, it works exactly as you suggest. When you select something that is a physical entity, you only get ontologies that contain physical entities. Now, your question is still good in that, some people will annotate against UniProt versus other people, the protein ontology, and then you're in trouble. So ultimately, we need to have a community and a list of sort of approved or sanctioned ontologies, but that's always a ticklish political business.
Yep.
So it's kind of out of scope of what David's presenting today, but it's in scope in terms of the long-term goals of this library.
Okay, the other thing I posted was the natural language to Sparkle query is really cool. It would be great if it could go natural language to annotation, right? Because really, if you think about it, it's the core process of naming things. And whether you're using that core process of naming things to generate annotation, or you're using that core process of naming things to generate a search query, you really want to go through the same process so that the search engine already knows about the same ontologies and relationships and can either strip it into natural language or vice versa, depending, but whether it's generating annotation or doing a search. The last thing is there a format export that's compatible with Google? Can you export as, I'm trying to think of what it is.
Yeah, I think I know what you mean, and no.
The British
That's the short
version.
answer.
What is Manchester syntax? So if you can export in Manchester syntax, that's a Google searchable syntax.
Huh. Well,
In
yeah,
RDF,
Manchester,
not
the Redlands library does support Manchester syntax.
Okay, so it'd be cool to make one of these, take one of these annotations and make it Manchester syntax and put it up somewhere and see if Google can find it.
Yeah.
Right, I mean, bottom line, somebody should not have to know where the repositories are.
No.
We should not need to know that. Plus the fact that the repositories are going to move in the future, right? An NIH funded repository has a lifetime of 6 or 7 years and then it's gone. Funding runs out, they go 2 years past the end of funding and the repository vanishes. So it would certainly be nice if a person didn't need to know what the repositories are and they could just do a Google search, and Google, Google indexes everything. We've done tests in the past where if you put a bqBio ID in a file, that makes that web file incredibly unique. bqBio : 1, 2, 3, 4, 5, 6, 7, 8 is really only going to find all the documents that Google knows about that actually are talking about that bqBio species. The problem is that Google just won't do XML. It's this historical thing of XML looks like HTML, and inside the angle brackets in HTML, there's nothing of value. The only thing that's inside the angle brackets are the font color is red and it's Arial and stuff like that. Well, I mean, there's two ways of doing XML, right? One is everything's in tag in the thing, and the other one is actually the information is never in the tag. It's open red or open color and then the word red and then close color, and so red is visible to Google, whereas if you do as a tag, it's not. Anyway. This looks really cool. I think it'd be really handy. I've got a project where I need to generate a whole bunch of annotations. And it looks like I would be able to write a Python script that could iterate over a library of images, for example, and you extract out whatever information's in the file name. These are all macaque monkey, they're all female, and generate a standard set of a starting point of a standard set of annotations.
So James, I should add, James, that you're absolutely right about the tedium of annotating models, which is why it's hardly ever done. We have two side projects that will be using this library. One is a smart editor that helps you create the annotations for you. And then another one is whether we could automate annotation in some way so that you don't have to do it manually. That's a more tricky problem.
Yeah.
So.
Yeah. I always had an idea of, could you extend Doxygen, the Doxygen annotation system for Python and C++ and such, where it lets you write the annotations for your code and you run it through the Doxygen filter and it pulls all your annotations out and makes a manual for your code.
Yeah.
If you could modify that a little bit and instead of putting in, annotations, instead of putting in, user documentation, put in annotations like this.
Yeah.
So in the middle of your code, you've got this RDF block that is this structured annotation. And there's a filter. Either you could search with Google or you'd run it through the Doxygen filter and it just pull out the RDF, all of the ontological descriptions and just.
Remember in this new way of doing things, the annotations are separated out from the model. And you just have pointers there's a separate annotation file, RDF file that has all the annotations. And then there are pointers.
Right, that's how Combine's done it.
Yes, that's how Combine
does it.
Yeah, well, that's what this supports.
But that's not the only way to do it. And even that way, the idea is to make it a little easier to generate bits and pieces of the annotation.
Yeah.
But I think this is really cool. I just need a web page to do it. That already knows. So I think I can say this is a chemical species, and oh, by the way, it's acetaminophen, and it goes off, and it's smart enough to know to go look at Gebby first.
Yeah,
always
you're right.
You have the capability of using the technology you want, but it wouldn't get used all that much because people really don't annotate all that much.
We could do that with the smart editor. I mean, the smart editor runs on Visual Studio Code, not on a browser. But one could imagine something maybe similar for a browser.
It would be great if it could, remember your remember your ORCID ID.
Yeah.
So every time I log in, it knows who I am. It already knows the ORCID ID. I don't, when I add an annotation, I don't have to tell what time it is. It just, fetches system time and puts the time on the annotation. But anyway, I think this library looks really cool. I'll have to see if I can play around with it a little bit.
Is this what Semantic SVML does? Is Semantic SVML a website to help you annotate? I don't know. I can't remember now.
There was a tool where it was kind of an XML editor.
Oh, right.
Embedded in the web browser. And it was actually pretty good because it knew if you said reaction, it knew you had products and things like that.
Right.
And so it had some knowledge.
Yeah, okay.
But it didn't do, I don't think it did external ontological annotations like linking it to Gebby.
Yeah. But a web browser thing might be of interest. Yeah.
And like I say, that's one of those things where it doesn't have to run on any other machine except for one. And you just run it as a web service for the world. Which makes coding much, much easier. You don't need a Linux and an Apple and a Mac and a PC. It's sitting on that old desktop that you got back of your office.
Yeah, it's an idea. Definitely an idea.
Okay, thanks.
Yeah, thanks, James.
I think we're pretty much out of time, but thank you, David, so much for making time for this talk. Thanks, Dr. Sluka, for generating a lot of discussion on it as well.
Cool, thanks.